Hi, good morning, everybody. Welcome back to our daily lectures on New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby. We were dealing on the subject of molecular biotechnology and the topic of regulatable promoters under the chapter of manipulation of gene expressions in prokaryotes. As I talk, you may please make screenshots and write notes. Go into the transcript and you can get the details. Please send in your queries and clarifications on the comment section. Kindly do not commercialize any aspects of the lecture or the notes. We wish to express our thanks and our courtesy to the authors Glick, Bernard, Jack, and Cheryl of the fourth edition of Molecular Biotechnology. Continuing on the subject, and in particular the topic of regulatable promoters, the effectiveness of deactivating a repressor protein and thereby activating transcription depends on the ratio of the number of repressors, protein molecules, to the number of copies of the promoter sequences. If there are too many repressor protein molecules, then it is difficult to induce transcription. Conversely, with too few repressor protein molecules, even when there are more repressor molecules than copies of the promoter transcription, occurs in the absence of induction in these cases. The promoters are said to be leaky in this case, or in such cases. Various means have been devised to keep these regulatory systems under complete control. For example, the repressor protein gene and the promoter that it regulates may be placed on two different plasmids that maintain different numbers of copies per cell. This arrangement maintains the appropriate ratio between the repressor protein and the promoter. Usually, the repressor gene is replaced on a low copy number plasmid that maintains about 1 to 8 copies per cell, and the clone gene with its promoter sequence is inserted into a high copy number plasmid that maintains about 30 to 100 copies per cell. Alternatively, the repressor protein gene may be carried as a single gene in the chromosomal DNA, an arrangement that keeps repressor protein levels low. In systems that use the LAC promoter, a mutant form of the LAC gene, LAC-L9, let me just magnify that for you, LAC-LQ, right, that produces much higher levels of the LAC repressor, is often used to decrease transcription leakiness under non-induced conditions. That transcription of a clone gene in the absence of inducer. What is this? DNA. However, this spacer region can in fact contribute to the strength of the promoter. When a portion of the spacer region from the E. coli LAC promoter was deliberately mutated, when a promoter of the spacer region from the E. coli LAC promoter was deliberately mutated and constructs that yield the most active promoters were selected, one construct displayed a greater than 40-fold increase in LAC promoter directed RNA synthesis in the absence of CRP, and an 8-fold increase in the presence of CRP, where CRP stimulated RNA synthesis tenfold. In the mutated promoter with increased activity, the 20 to 13 region of the promoter was altered from a GC-rich 7 out of 8 BP to an AT-rich 7 out of 8 pp region. Moreover, when the 20 to 30 region was excised from the mutated lac promoter and inserted into the P8 promoter from bacteriophage lambda, this transcription was enhanced twofold. This is important because while the lac promoter is normally relatively weak in the absence of CRP, the PR promoter is one of the strongest promoters in E. coli. Thus, by substituting an AT-rich region for a GC-rich region and possibly enabling the promoter region to form a stronger complex with RNA polymerase, the intrinsic strengths of various promoters, even strong ones, may be increased. These altered promoters may be utilized as parts of an expression vector. Increasing protein production. Increasing protein production. Plasmid PCP3, figure 6.6, was created in an effort to obtain the highest possible level of foreign protein production in a recombinant E. coli strain. This plasmid contains a strong P1PL promoter, the beta-lactamase gene ampicillin resistance gene as a selective marker, a multiple cloning sequence immediately downstream from the promoter, and a temperature-sensitive revision of DNA replication that increases the plasmid's copy number 5 to 10-fold when the growth temperature is increased to 42 degrees centigrade. E. coli cells that carry the plasmid PCP3 are first grown at 28 degrees centigrade and then shifted to 42 degrees centigrade. At the lower temperature, the CL repressor, which is integrated into the host E. coli chromosomal DNA, is functional. The PL promoter is turned off and the plasmid copy number is normal, about 60 copies per cell at the highest temperature. At the higher temperature, the temperature-sensitive CL repressor is inactivated, the PL promoter is active, and the plasma copy number increases to around 600 copies per cell. These properties made PCP3 a particularly effective expression vector when the gene for the enzyme T4DNA ligase is inserted into the multiple cloning site of PCP3. About 20% of the cellular protein produced at 42 degrees centigrade is T4DNA ligase. This level of expression is much higher than that for even the most abundant native E. coli proteins, such as the elongation factor EFTU, which have an expression level of about 2%. Large-scale systems. In small culture vessels 1 to 5 liters, induction is readily achieved either by shifting the temperature or by adding a chemical inducer. Figure 6.6 is a linear representation of E. coli expressor vector PCP3 with a temperature-sensitive origin of DNA replication ORI-TH that causes an increase in the plasmid copy number at 42 degrees centigrade and a PL promoter controlled by a temperature-sensitive repressor protein, a multiple cloning site MCS, and an ampicillin resistance AMP gene. Figure 6.7 shows E. coli cells that contain a plasmid with a temperature-sensitive origin of DNA replication. It contains a limited number of plasmid copies at 28 degrees centigrade, which is greatly amplified when the temperature shifts to 42 degrees centigrade. So here 20 to 200 liters and industrial size greater than 200 liters bioreactors are used. However, a shift in temperature requires time, 30 to 60 minutes, and energy, both of which can be costly. Similarly, the cost of a chemical factor inducer, such as IPTG, that is required for the expression of a clone gene in a large-scale bioreactor can make the overall process uneconomical. To overcome some of the problems associated with the use of the PL promoter for large-scale fermentations, a two-plasmid system has been developed. The CL repressor was placed under the control of the GRP promoter and inserted into a low copy number plasmid. The use of a low copy number plasmid ensures that excess CL repressor molecules are not produced. A second plasmid carries a clone gene under the control of the PL promoter. As shown in figure 6.8a, the TRP promoter is turned on in the absence of tryptophan, so the CL repressor protein is synthesized and the PL promoter is turned off. In contrast, as shown in figure 6.8b, the TRP promoter is often turned off in the presence of tryptophan, so the CL repressor protein is not synthesized and the PL promoter is fully active. With these two plasmid systems, cells can be grown on an inexpensive medium consisting of molasses and casein hydrolysate, which contains only very small amounts of free tryptophan, and then induced to express the clone gene by the addition of tryptone to the medium. Tryptone contains enough free tryptophan for efficient induction of transcription. In trial runs of the system, cloned beta-galactosidase and citrate synthase genes after induction by addition of tryptone to the medium represented 21 and 24% of the cellular protein, respectively. Thus, this system provides a potentially inexpensive means of producing proteins from recombinant microorganisms on a large scale. Several considerations may limit the choice of promoters for the large-scale production of foreign proteins. Chemical inducers can be costly, toxic, or difficult to remove. Thermal induction promoters may induce the production of heat shock proteins, including proteases. Nutrient promoters limit the types of media that can be used for cell growth and induction, and oxygen-regulated promoters often have significant basal levels of activity as a consequence of the inherent difficulty in precisely controlling dissolved oxygen levels in the growth medium. On the other hand, promoters that are induced when cells enter stationary phase may be useful in the design of expression vectors that are useful for large-scale applications. In E. coli, the housekeeping RNA polymerase sigma factor sigma L3 as shown. Let me just magnify that. Sigma D recognizes and binds to a well-characterized consensus DNA sequence at approximately 10 and 35 BP upstream from the site where transcription occurs. This is figure 6.8, which shows a dual plasmid system for controlling lambda PL, promoted by regulating the CL repressor with tryptophan via tryptophan promoter. TRP is inserted next to the CL gene on one plasmid, and the PL promoter is placed adjacent to the clone gene on a second plasmid. The wavy arrows denote transcription A with no tryptophan in the medium. The CL gene is transcribed and translated, and the CL repressor protein binds to the PL promoter, thereby blocking the transcription of the clone gene B. In the presence of tryptophan, the CL gene is suppressed and no CL product is made, and the clone gene is transcribed and translated, whereby it is initiated, and as shown in figure 6.9, the stationary phase of sigma factor, sigma as shown here, sigma S, recognizes a similar but not identical sequence of nucleotides in the same region upstream of the start of transcription. Despite the similarities between the two types of promoter sequences, the 35 region appears not to be important for the functioning of the known stationary phase promoters. Good morning, and the time is 6 o'clock Indian Standard Time. The time is 6 p.m. Indian Standard Time. Despite the similarities between these two types of promoter sequences, the 35 region appears not to be important for the functioning of the known stationary phase promoters. Assuming that the sequence around the 35 region affect promoter activity, in one study, researchers generated more than 150 different stationary phase promoters in which the DNA sequences upstream of the 10 consensus sequence were partially randomized. These workers found that a number of these synthetic promoters had 3 to 4 times the level of activity of naturally occurring stationary phase promoters and had no or only a very low level of background gene expression during exponential growth. Thus, it is possible to use these promoters as part of an expression vector. In this case, the cells would be grown to a high density without expressing a foreign gene of interest, and as the cells entered stationary phase, gene expression would be rapidly and efficiently induced. Despite its success on a laboratory scale, the effectiveness of the system remains to be demonstrated on a large scale. The figure 6.9 shows consensus sequences of sigma D dependent top and dependent sigma S dependent bottom promoters. The 10 region is shown, including the 13 residue, since it is important for sigma S binding recognition. The 80 rich region downstream of the 10 region and the 8 WT sequence contribute to sigma selectivity and indicates any one of the A, C, G, or T, whereas W indicates A or a T at the position. The 10 and 35 residue are indicated by an asterisk. This courtesy to Mixed, which is modified from Mix et al. At BIOTECHNOLOGY. Thank you. Now, on to the next topic. Expression in other microorganisms. E. coli is not necessarily the microorganism of choice for the expression of all proteins. However, our understanding of the genetics and molecular biology of most other microorganisms is not as well developed. Unfortunately, there is no one vector or promoter repressor system that gives optimal levels of gene expression in all bacteria or even in all gram-negative bacteria. Fortunately, many of the strategies that have been developed for E. coli are also useful with a variety of other microorganisms. With this in mind, the abilities of various promoters to support transcription in other gram-negative bacteria have been tested. In one study, a set of plasmid expression vectors containing either the LAC or TAC enum from the plasmid expressions from the neomycin resistance gene, or S1 from the ribosomal protein S1 gene of Cynorhizobium meliloti. Promoter was constructed. The expression of beta-galactosidase under the control of each of these promoters was examined, as shown in Table 6.1. The results indicated that 1. All of the promoters were active to some extent in each of the bacteria tested. 2. The TAC promoter was the most active promoter in E. coli and the least active promoter in the other bacteria. And 3. NM was the second least active promoter in E. coli and the most active promoter in the other bacteria. Clearly, even though gram-negative organisms may utilize similar DNA sequences to promote transcription, the best promoter for EUs in a particular organism is not necessarily the one that is most efficient in E. coli. Nevertheless, depending on the application known, E. coli promoters may be satisfactory for regulating the expression of clone genes in other gram-negative bacteria. Lactic acid bacteria, example Lactococcus spp, are widely used in the production of foods such as cheese and yogurt. Genetic manipulation of these bacteria is desirable to increase yields or add to the quality of the product. However, any changes must not affect the production process, product palatability in appearance or other features. For these reasons, it is not acceptable to modulate gene expression by adding chemical inducers or significantly modifying process conditions such as temperature. In addition, the range of attainable foreign gene expression is intrinsically limited when only the available constitutive promoters are used. To overcome this limitation, a plasmid library of synthetic promoters for Lactococcus lactis was constructed in which the 10 and 35 regions of each promoter were the same, but the sequences of the intervening spacer nucleotides were randomized. To assay the strengths of the individual constructs, each synthetic promoter was used to control the expression in the related bacterium Leuconostoc lactis or the LAC-M genes, which together encode beta-galactosidase. To quantify the strengths of the various promoters, the beta-galactosidase activity from each construct was measured. Of the 36 different constructed promoters that were tested, the most active promoter was approximately 7,000 times stronger than the least active promoter. DNA sequence analysis revealed that the least active promoters all had changes in the 10 or 35 consensus region. Of the promoters in which the 10 or 35 consensus region remained intact, there was a 400-fold variation in promoter strength. These results indicated that the sequence of the spatial region is important for promoter activity. With this panel of promoters, it is possible to fine-tune the expression of different genes that are introduced in L. lactis and by extension the principle the expression of genes introduced in other organisms as well. Before we go to fusion proteins, we have a figure to be an illustration, or a table in fact. It shows beta-galactosidase activity expressed by gram-negative bacteria carrying a plasmid vector with the E. coli LAC-Z gene and a heterologous promoter. Right, on to the subject of fusion proteins. Now we deal with the topic of fusion proteins. Often foreign proteins, especially small ones, occur in minute quantities. When they are produced in heterologous quantities or heterologous host cells, this apparently low level of expression is in many instances actually due to degradation of the foreign protein. One way to solve this problem is to engineer a DNA construct that encodes a target protein that is in frame with its stable host protein. This combined single protein, which is called a fusion protein, protects the clone gene product from attack by host cell proteases. In a number of studies, proteins synthesized from clone genes have been found to be resistant to degradation when they are part of a fusion protein, whereas when they are expressed as separate proteins, they are susceptible to degradation by proteolytic enzymes proteolysis. In general, fusion proteins are stable because the target proteins are fused with proteins that are not especially susceptible to proteolysis. Fusion proteins are constructed at the DNA level by ligating a portion of the coding regions of two or more genes. In its simplest form, a fusion vector system entails the insertion of a target gene or gene segment into the coding region of a cloned host gene. Knowledge of the nucleotide sequences of the various coding segments that are joined at the DNA level is essential to ensure that the ligation product maintains the correct reading frame. If the combined DNA has an altered reading frame, that is a sequence of successive codons that yields either an incomplete or an incorrect translation product, then a functional version of the protein encoded by the cloned target gene will not be produced. Many strategies have been devised to ensure that a proper reading frame is achieved. Uses of fusion proteins. For some applications, a fusion protein can be a satisfactory end product. For example, a specific antigenic site that is required in large amounts and is part of a fusion protein may be used for research or diagnostic purposes. Figure 611 shows a fusion protein cloning vector The plasmid contains an ampicillin resistance AMP-R gene, empty, let me just magnify that. Yeah, it's AMP-R. Gene as the selectable marker. A DNA sequence encoding the N-terminal segment of the E. coli outer membrane protein OMPF, a restriction endonuclease site, ABCL for cloning, and a truncated beta-galactosidase gene, LAGZ. The cloned gene is inserted into the ABCL site after transcription and translation. A trihybrid protein is produced consisting of OMP-F target protein LAGZ. AMP-TG is shown in the illustration. It's cut off with ABCL ligates to give the AMP gene. Alright, go on to the. As long as the stabilizing protein does not interfere with the correct folding of the antigenic site. In this case, the fusion protein can be used as an antigen, and any antibodies that are directed against the stabilizing protein can be removed by absorption with this protein alone, thus leaving in the antiserum only those antibodies that bind to the targeted protein sequence. In one instance, a fusion cloning vector that included the 5' terminal segment of the E. coli OMP-F gene, which directs the synthesis of an outer membrane protein, and the portion of the E. coli LAGZ beta-galactosidase gene was constructed and used to generate antibodies against selected target proteins. Figure 611 shows that the OMP-F gene segment contributed the signals for the initiation of both transcription and translation and for secretion of the fusion protein. Even though the truncated LAGZ gene lacks the codons for the first 8 amino acids, the shortened protein encoded by this gene fragment is still enzymatically active. This form of the enzyme B-galactosidase is able to function with almost any peptide fused to its N-terminus. The LAGZ gene was cloned in the vector at a location that put it in an altered reading frame with respect to the OMP-F leader sequence. Therefore, no functional beta-galactosidase was produced. However, any clone target DNA that had both OMP-F and LAGZ in frame would promote a 3-part hybrid protein that comprised a portion of the OMP-F amino acid sequence. That protein encoded by the clone target gene and the functional C-terminal portion of the beta-galactosidase, whose activity is readily visualized on plates. Such a hybrid protein can be used either as an antigen to produce antibodies that will cross-react with the protein encoded by the clone gene, or as a means of producing large amounts of small important portions of specific proteins. In addition to reducing the degradation of polypeptides, a number of fusion proteins have been developed to simplify the purification of recombinant proteins. Table 6.2 shows that this approach is useful for purification of proteins expressed in either prokaryotic or eukaryotic host organisms. Some protein fusion systems used to facilitate the purification of foreign proteins in E. coli are shown here in table 6.2. Fusion partner, the size, the ligand, and the elution conditions, along with the host organisms. The fusion partners will be ZZ, histidine tail, strep tag, pinpoint, maltose binding protein, GST, flag, polyarginine, C-mic, S-tag, calmodulin binding peptide, cellulose binding domain, chitin binding domain, and SBP tag. Now, fusion proteins are constructed at the DNA level by ligating a portion of the coding regions of 2 or more genes. In its simplest form, a fusion vector system entails the insertion of a target gene or a gene segment into the coding region of a cloned host gene. Knowledge of the nucleotide sequence of the various coding segments that are joined at the DNA level is essential to ensure that the ligation product maintains the correct reading frame. If the combined DNA has an altered reading frame, that is a sequence of successive codons that yields either an incomplete or an incorrect translation product, then a functional version of the protein encoded by the cloned target gene will not be produced. Many strategies have been devised to ensure that a proper reading frame is achieved. Uses of fusion proteins. For some applications, a fusion protein can be a satisfactory end product. For example, a specific antigen site that is required in large amounts and is part of a fusion protein may be used for research or diagnostic purposes. Figure 611 shows that. Now, figure 611 shows the fusion protein cloning vector. The plasmid contains an ampicillin resistance AMPF gene as a selectable marker, a DNA sequence encoding the N-terminal segment of the E. coli outer membrane protein or MPF, a restriction endonucleotide ABCL for cloning, and a truncated beta-galactosidase gene LAGZ. The clone gene is inserted into the ABCL site. After transcription and translation, a tribrid protein is produced consisting OMPF target protein LAGZ. The illustration is here for you to see. Please take screenshots and use them to study. Now, as long as the stabilizing protein does not interfere with the correct folding of the antigenic site, in this case, the fusion protein can be used as an antigen, and any antibodies that are directed against the stabilizing protein can be removed by absorption with this protein alone, thus leaving in the antiserum only these antibodies that bind to the targeted protein sequence. In one instance, a fusion cloning vector that included the 5' terminal segment of the E. coli OMPF gene, which directs the synthesis of an outer membrane protein, and a portion of the E. coli LAGZ beta-galactosidase gene was constructed and used to generate antibodies against selected target proteins. The OMPF gene segment contributed signals for the initiation of both transcription and translation and for secretion of the fusion protein. Even though the truncated LAGZ gene lacks the codons for the first 8 amino acids, the shortened protein encoded by this gene fragment is still enzymatically active. This form of the enzyme B-galactosidase is able to function with almost any peptide fused to its N-terminus. The LAGZ gene was cloned on the vector at a location that put it in an altered reading frame with respect to the OMPF leader sequence. Therefore, no functional beta-galactosidase was produced. However, any clone target DNA that had both the OMPF and LAGZ in frame would produce a 3-part hybrid protein that comprised a portion of the OMPF amino acid sequence, the protein encoded by the clone target gene, and the functional C-terminal portion of beta-galactosidase, whose activity is readily visualized on plates. Such a hybrid protein can be used either as an antigen to produce antibodies that will cross-react with protein encoded by the clone gene, or as a means of producing large amounts of small, important portions of specific proteins. In addition to reducing the degradation of polypeptides, a number of fusion proteins have been developed to simplify the purification of recombinant proteins. This approach is useful for purification of proteins expressed on either prokaryotic or eukaryotic host organisms. Table 6.2 shows that the fusion partners, size, and ligands. Here we have ZZ going for 14K kilodaltons and immunoglobulin G. And the others will be histidine tail, the strep tag, pinpoint, maltose binding protein, GST, flag, polyarginine, EY, MYC, S tag, and calmodulin binding peptide, cellulose binding domain, chitin binding domain, SPB tag. Right. And the ZZ, a fragment of Staphylococcus aureus protein A, strep tag, a peptide with affinity for streptavidin, pinpoint, a protein for fragment that is biotinylated and binds with the streptavidin. Now you have this table with these proteins and peptides. Well, just to get a little idea of it, ZZ, a fragment of Staphylococcus aureus protein A, and strep tag, a peptide with affinity for streptavidin. Pinpoint is a protein fragment that is biotinylated and binds with streptavidin. GST, glutathione transferase, and flag, a peptide recognized by enterokinase. CMYC, a peptide from a protein that is overexposed in many cancers. S-tag, a peptide fragment of ribonuclease RNase A. SPB, streptavidin binding protein. a peptide with affinity for streptavidin, specifics, a cation exchange, or a cephadics, a cation, and that exchange, which is resin composed of sulfur-propyl groups. And attached to cephadic beans. Right. Well, now this is an example of a vector that contains the human interleukin-2 gene joined to DNA encoding. The marker peptide sequence, as shown here, has the dual function of reducing the degradation of the expressed interleukin-2 gene product and then enabling the product to be purified. Interleukin-2 is a biological factor that stimulates both T cell growth and B cell antibody synthesis. Following expression of this construct, the secreted fusion protein can be purified in a single step by immunoaffinity chromatography, in which monoclonal antibodies against the marker peptide have been immobilized on a polypropylene or other solid support and act as ligands to bind the fusion protein. Because the marker peptide is relatively small, it does not significantly decrease the amount of host cell resources that are available for the production of interleukin-2. Thus, the yield of interleukin-2 is not affected by the concomitant synthesis of the biomarker peptide. In addition, while the fusion protein has the same biological activity as native interleukin-2 to satisfy the government agencies that regulate the use of pharmaceuticals, it is still necessary to remove the marker peptide if the product is to be used for human immunotherapy or other medical purposes. In the system, the marker system may be specifically removed by treatment of the fusion protein with bovine intestinal enterokinase, which is a highly specific protease despite its name. In many instances, antigen-antibody complexes that form during the immunoaffinity process are difficult to separate without the use of denaturing chemicals. As a result, it has become very popular to generate a fusion protein containing 6 or 8 histidine residues attached to either the N or C terminal end of the target protein. The histidine tag protein, along with the other cellular proteins, is then passed over an affinity column of nickel nitrilotriacetic acid. The histidine tag protein, but not the other cellular proteins, binds tightly to the column. The bound protein is eventually eluted from the column by the addition of imidazole, the side chain of histidine. With this protocol, some cloned and overexpressed proteins have been purified up to 100-fold with greater than 90% recovery in a single step. In addition, this system can be utilized to purify denatured proteins, for example, following solubilization of inclusion bodies and before the solubilized proteins are renatured. Cleavage of fusion proteins. Depending on its use and its end use, it may be undesirable to produce a fusion protein as the final product. For example, the presence of the host protein segment makes most fusion proteins unsuitable for clinical use and may affect the biological functioning of the target protein. In addition, fusion proteins require more extensive testing before being approved by regulatory agencies. Figure 6.12 shows a schematic representation of the genetic construct used to produce a secreted fusion protein consisting of a marker peptide and interleukin-2. The amino acid sequence of the marker peptide is shown. This is the continuation of the marker peptide. And the figure 6.13 shows an immunoaffinity chromatographic purification of a fusion protein. An antibody that binds to the marker peptide of the fusion protein anti-marker peptide antibody is attached to a solid polypropylene support. The secreted proteins are passed through the column containing the bound antibody. The marker peptide portion of the fusion protein is bound to the antibody and the other proteins pass through. The immunopurified fusion protein can then be selectively eluted from the column by the addition of pure marker peptide. And agencies such as the US Food and Drug Administration have been noting this. And the strategies have been developed to remove the unwanted amino acid sequence from the target protein. One way to do this is to join the gene for the target protein to all or a portion of the gene F for another protein, the stabilizing fusion partner, with oligonucleotides that encode short stretches of amino acids that are recognized by a specific non-bacterial protease. The oligonucleotide linkers that code for the protease recognition site may be ligated upstream of the clone gene so that the linker peptide will be synthesized at the N-terminal end of the target protein before the construct is inserted into. A fusion expression vector, for example, an oligonucleotide linker encoding the amino acid sequence, as shown here, can be joined to the clone gene following synthesis and purification of the fusion protein. A blood coagulation factor called X can be used to release the target protein from the fusion partner because factor XA is a specific protease that cleaves peptide bonds uniquely on the C-terminal side of the shown sequence. Moreover, because this peptide sequence occurs rather infrequently in native proteins, this approach can be used to recover many different cloned gene products. The proteases most commonly used to cleave a fusion partner affinity tag from a protein of interest are enterokinase, tobacco H virus protease, thrombin, and factor XA. However, following this cleavage, it is necessary to perform additional purification steps in order to separate both the protease and the fusion protein from the protein of interest. In addition, proteases may cleave the protein of interest at unintended sites, and the cleavage reaction may not go to completion, leaving a portion of the protein of interest still attached to its fusion partner. One way around this problem is through the use of self-splicing inteins. An intain may be defined as the internal segment of a protein. Okay, and under specific conditions, under specific conditions, catalyzes its own cleavage into two separate polypeptides. Of the more than 100 known inteins, the majority contain a cysteine or a serine residue at the N-terminal side of an asparagine residue, with cleavage occurring between these two amino acids. When the N-terminal residue at the cleavage site is sustained, protein chain cleavage may be initiated by the addition of a sulfhydryl reagent, such as dithiothiotol. This approach typically includes a gene fusion with the gene of interest encoding the target protein. An intein and a protein tag. One inexpensive way to commercialize the system is to utilize a chitin binding domain as the protein tag. In practice, a mixture of cellular proteins is passed through a chitin column to which only the fusion protein binds. The rest of the protein passed through the column. The column is treated with dithiothiotol. The protein of interest is cleaved at the intein junction and eluted. The inexpensive used column is discarded. This system has been used to purify CRE recombinase, a tool for chromosome engineering, alpha-1 antitrypsin, a therapeutic protein, and basic human fibroblast growth factor, a potential therapeutic protein. Surface display, a specialized protein synthesis that have been devised for screening complementary DNA, cDNA libraries that contain very large numbers of different clones, sometimes up to 5 to 10 raised to 10 proteins that are encoded by rarely occurring cDNAs. Generally, these libraries, cDNA molecules, are cloned into a surface protein, filament protein, or pilus protein, gene of either a filamentous bacteriophage such as M13 or a bacterium. After transcription translation, the fusion protein is incorporated into a surface structure of the bacteriophage or bacterium, where it can be identified by an immunological assay. More specifically, fusions are often made with the protein P3 gene from bacteriophage M13. Protein P3 is normally found at the tip of this tubular bacteriophage and is responsible for initiating phage infection of E. coli by binding to F. pili. A plasmid that contains a small piece of M13 DNA that allows the plasmid phagement to be packed in vitro into M13 phage particles, a protein P3 gene under the control of a regulatable bacterial promoter, such as the E. coli lac promoter. And the cloning site near the 5' end of the P3 gene for insertion of a cDNA or complementary DNA or other coding sequence is constructed. The express target protein is fused to bacteriophage M13 protein, M13 protein, P3 near its N-terminus. After M13 replication, in E. coli cells, the plaques are assayed immunologically using antibodies that detect the presence of the target protein. Recombinant phagements isolated from the positive plaques can be used as a source of target DNA. This is an extremely powerful selection system that has the capability of finding cDNAs for very rarely expressed but important proteins. Finally, it is also possible, although less straightforward and therefore less common, to create recombinant phage in which the target peptide is fused to protein P8, the bacteriophage code protein. An alternative to the phage surface display of proteins described above is the use of libraries with bacterial surface structures composed of fusion proteins that can be screened for clones that carry specific coding sequences. To export proteins to the surface of a gram-negative bacterium such as E. coli, fusions between the genes for the target protein and for an outer surface protein are created. Bacterial fusion partners that have been used in these types of constructs include outer membrane protein, OMP-A, and peptide glycan-associated lipoprotein PAL from E. coli, as well as Pseudomonas aeruginosa, outer membrane protein OPRF, with most bacterial surface fusion proteins, the target protein is located at either the N or C terminus of the fusion protein. However, in some instances, short stretches of its target protein can be expressed in the middle of the fusion partner. In addition to facilitating the screening of large cDNA libraries, surface display proteins can provide an effective means of overexpressing. This is a figure, which is number 6.16, which is expressive of the recombinant bacteriophage M13 displaying a fusion protein consisting of the target protein and a phage protein P3. Take a screenshot if you can. And figure 6.17 shows a fusion protein between bacterial outer surface protein and a foreign target protein joined together to either the N or C terminus or foreign peptides inserted into the surface exposed loops. In both cases, the foreign peptides, the protein is directed to the outer surface of the bacterial cell. Peptides are proteins, for example, in one study, the amino acid repeating epitope antigenic determinant, as shown here, of a protein from the parasite Plasmodium falciparum, the causative agent of malaria, was inserted into the regions that encoded surface exposed loops of the major outer membrane protein from P. aeruginosa, OPRF. Whole bacterial cells expressing this fusion protein reacted positively when challenged with monoclonal antibodies against P. falciparum. It may therefore be possible to use some surface display fusion proteins as vaccines. Translation expression vectors. Putting a cloned gene under the control of a regulatable strong promoter, although essential, may not be sufficient to maximize the yield of the cloned gene product. Other factors such as the efficiency of translation and the stability of the newly synthesized cloned gene protein may also affect the amount of product. In prokaryotic cells, various proteins are not necessarily synthesized with the same efficiency. In fact, they may be produced at very different levels, up to several hundredfold, even if they are encoded within the same polycistronic messenger RNA. mRNA differences in translation efficiency and in transcriptional regulation enable a cell to have hundreds or even thousands of copies of some proteins and only a few copies of others. In part, the molecular basis for differential translation is the presence of a translation initiation signal called ribosome binding site transcribed mRNA. A ribosome binding site is a sequence of 6 to 8 nucleotides in mRNA that can base pair with a complementary sequence on the RNA component of the small ribosomal subunit. Generally, the stronger the binding of the mRNA to the ribosomal RNA, the greater the efficiency of translational initiation. For this reason, many E. coli expression vectors have been designated to ensure that the mRNA of a cloned gene contains a strong ribosome binding site. In effect, this means that heterologous prokaryotic and eukaryotic genes can be translated readily in E. coli. However, certain other conditions must be satisfied for this approach to function properly. First, the ribosome binding sequence must be located at a precise distance from the translational start sequence or transcodon of the cloned gene. At the RNA level, the translational codon is usually AUG. At the DNA level, the strand that has the ATG sequence is called the coding strand, and the complementary strand, which acts as the template for transcription, is the non-coding strand. By convention, a start codon at the DNA level is designated ATG. Second, the DNA sequence that includes a ribosome binding site through the first few codons of the gene of interest must not contain nucleotide sequences that have regions of complementarity and therefore, after transcription, can fall back, that is, form intrastrand loops, figure 6.18, thereby thwarting the interaction of the mRNA with the ribosome. The local secondary structure of the mRNA, which can either shield or expose the ribosome binding site, determines the extent to which the mRNA can bind to the appropriate sequence in the ribosome and initiate translation. We're going back to a milestone note. This is the attack promoter, a functional hybrid derived from the TRP and LAC promoters, courtesy H.A. DeBoer, L.J. Comstock, and M. Vassar, and Proctor National Academic Scientific USA, 1993, credits to them. DeBoer and his colleagues began their efforts to construct the attack promoter with the idea of combining portions of two different strong and regulatable promoters to create an even stronger promoter that would direct very high levels of foreign gene expression. When they undertook their studies, although the DNA sequence of a number of prokaryotic promoters, mostly from E. coli, were known, the precise features that enable a promoter to be efficient at directing transcription were not well understood. It was not known that almost all mutations that affect the strength of a prokaryotic promoter were found in either the 10 region or the 35 region, which approximately 10 or 35 base pairs upstream of the mRNA transcription start site, respectively. This lecture will end now in a minute, and will continue. Portions will be continued to the next lecture. Please stay tuned, right? And more about early mutations that made an existing promoter more like the consensus sequences. For each of these regions, that is the 5' TARA 3' for the 10 region and the 5' TDGACA 3' for the 35 region increased the strength of the promoter. The consensus sequences that had been deduced by comparing the DNA sequences that had been deduced by comparing the DNA sequence of all known promoters and determining which nucleotides occurred. Most often, Debo and his colleagues also knew the LAC-UB5 promoter, which is a stronger variant of the LAC promoter.